group of digestible and non-digestible. These are very important. We are not going to devote any time right now to not digestible carbohydrates. However, so-called dietary fibers based on cellulose, which is built from the same monomer, glucose, as the digestible are based. However, there is a different bond between the glucose units, a beta bond, and we are not able, beta glycosidic bond, that we are not able to hydrolyze, and we are able to hydrolyze the alpha glycosidic bond, that is the major bond in macromolecular digestible and smaller molecular like disaccharide digestible carbohydrates. So among these polysaccharides that we most commonly use in diet is starch, present in many plants like potatoes, like rice, etc., and glycogen, which is present in meat. Glycogen is present in meat that we eat, so I will tell a little more about that later on. So these are two major categories, and now digestible, because non-digestible are, as I mentioned, very important for our health, for the appropriate functioning of gastrointestinal tract. However, they are not absorbed because they are not digested. They are not hydrolyzed in the gastrointestinal tract. Digestible are converted to monosaccharides from which they are built, which compose them. So we have glucose, galactose, fructose, and another simple pentoses, etc., that we will discuss briefly later on. They are very important, but the major of them, the most important monosaccharide, hexose, is aldose, called glucose, as you already know from chemistry part of that problem, which was discussed a little earlier. So the major role of glucose is to deliver energy in a process of oxidation that is called glycolysis in general. So it is the major way to reach energy, to convert the energy in which they are rich into the energy that is useful in the living organism, living animal organism. On the other hand, not only, by the way, not only animal, but also bacteria, they also use glucose preferentially. So then glucose can be also alternatively oxidized, and both in respect to this type of the process that we are going to discuss in more details later, pentose phosphate pathway, as well as based on glycolytic intermediates. They serve to synthesize numerous structures in our organism, as we will see later, regarding amino acid, lipids very soon, and later on nucleotides, many various structures based on the intermediates from the glycolysis or from the pentose phosphate pathway. If we have excess of that compound, so we don't have to immediately oxidize it to get energy, then we can store it. That's a very important aspect, because we have to have, as you will see later on, we have to have certain amount of glucose present in organism. But the major form in which it is present, it is stored in our organism, is a macromolecular structure, glycogen. So very briefly, simple diet sugars are, as you see here, I won't read it, are usually rich in sucrose or saccharose, as some people say saccharose, some sucrose. This is the disaccharide built out of fructose and of glucose. Roughly, this is not the recommendation of any diet. This is just due to how we see how our organism functions. Roughly a little more than 50% of energy that we gain every day should origin from catabolism of carbohydrates, the best macromolecular form. That's why I underline them, because those macromolecular are not so risky in terms of the immediate negative consequences. So just to mention them, and you will face this problem as a future physician many times during the clinics as well. These are dental caries. This is obesity, which is so common nowadays. This is the problem of malnutrition, but not in quantity, but in quality, because it's just not enough to feed someone with the excess of simple carbohydrate. And cardiovascular diseases, which are strictly linked to obesity. So we not recommend, based on knowledge of the metabolism, to consume excessive amount of simple sugar, but rather, if you like sugar, if you like carbohydrates, rather the complex sugars, complex macromolecular sugars. Okay. Now let's go to the how we digest. So the diet that is composed of starch, which is a macromolecular plant carbohydrate or polysaccharide, the glycogen, which is animal origin polysaccharide, also both are built of glucose, and in both cases glucose is bound by alpha glycosidic bonds, 1,4 and 1,6. Saccharose, as I mentioned, or sucrose, as I mentioned, is built out of the fructose and glucose, and lactose, galactose, and milk sugar, so-called table sugar. Finally, some glucose and some fructose in fruits, mostly where it is present, in honey, for example, etc. So if we have the average diet, then we eat those type of sugars in diet. And then first touch is in mouth, because the salivary glands produce amylase, alpha amylase. This amylase alpha, because as I mentioned earlier, we can only digest the alpha glycosidic bond. This amylase decreases the size of those polysaccharides. So produce out of polysaccharides limited dextrins and limited dextrins. It's the size of that dictate the name of that, but these are oligosaccharide, larger or smaller oligosaccharide built of mostly glucose, because these are starch and glycogen are the macromolecular polysaccharides. Then in stomach, nothing. So this is just initial process that wet them a little, give some water to that, makes the mixture that gets into the stomach. In stomach, nothing happens. It passes through, and then it goes to the duodenum and slowly moves, being still now continuously degraded by pancreatic alpha amylase. This is, for short we say amylase, but this is again alpha amylase, but it's pancreatic, not saliva, but pancreatic alpha amylase, the most important enzyme responsible for digestion of carbohydrates in gastrointestinal tract. So it still continues to decrease the size of those structures and release saccharides, like those that were listed above. So glucose, galactose, fructose. And these are the majority of them, out of which 90% at least is glucose. So that's why we sometimes say that metabolism of carbohydrates is mostly metabolism of glucose, for simplicity. And on the ileum, on the surface of enterocytes, they are disaccharadases. The enzymes like glucoamylase, lactase, which converts lactose to galactose and glucose, saccharase, that was fructose and glucose, etc. Isomaltase, which is the iso form of maltase. Maltase are two consecutive glucose unit bound by alpha glycosidic bond 1,4. So this is the alternative 1,6, etc. So these are enzymes that finalize the process of digestion, and basically at the end of the whole process, down there in small intestine, we have basically the mixture of monosaccharides, out of which, as I mentioned, around 90% is glucose. And now those monosaccharides are absorbed because they were already degraded to the form, to the size that can enter cell. So the enterocytes, so they are absorbed from gastrointestinal tract, from small intestine, and the major two ways. First of those ways is the symport of glucose and galactose goes through the same transporter that is called the S for intestine. For gastrointestinal tract, it's a little different name, is as glucose transporters. Glucose transporter 1, and this is responsible for absorption, for intake of glucose and also galactose, but together with sodium ion. However, to have the higher concentration of sodium ion, as you see here in a lumen of intestine, is 0.14 molar, cellular is just only 0.03 molar, so it's much lower. Just to reach that difference in concentration, we have to pump, and this electrogenic ATP sodium potassium generates the gradient of sodium ion that is the driving force to bind to this transporter glucose or galactose and transport it into the cell, to the cytoplasm of enterocyte. And in case of fructose, I would just briefly mention, there is so-called facilitated diffusion. So there is no need for sodium ion to be co-transported. There is no simple facilitated diffusion. So there is a transporter that allows to pass, but just due to the gradient of fructose. Now, we focus mostly on glucose, as I mentioned, because, as I told you, glucose is the major monosaccharide and building blocks of practically exclusive building block of polysaccharides. Therefore, we can focus on and discuss the glucose metabolism. We will later on, at the end of our lectures regarding carbohydrates, we'll briefly come back for a moment to galactose and fructose. But glucose is the major metabolism of carbohydrates in our organism. And now, on the apical side, it is just transported by uniport due to the gradient of concentration. There is higher concentration due to the absorption from gastrointestinal tract, and then import passes take and transport it to the portal vein, and this way it goes through the liver and goes to the blood circulation. And that's the way how we increase the concentration of glucose upon the digestion in gastrointestinal tract after the meal. Now, Glucose enters cells always by facilitated transport, like fructose from lumen of gastrointestinal tract, from intestine. However, here we have the blood, and glucose from blood is taken up by the so-called glucose transporters. I will focus for a moment on those glucose transporters to say a little more. And it moves always from the compartment where the higher concentration to lower. So that way, when we increase the concentration of glucose in blood, then it is taken by those glucose transporters and gets into the cell. Those transporters, some at least of them, are regulated by insulin. That's very important. We will come to the role of insulin in metabolism of carbohydrates, and not only, but the metabolism in general later on. But it's just to mention at the beginning, and the role how insulin interferes with the process is that, as you see, some of those glucose transporters, like in red blood cells, does not depend on insulin. Red blood cells has to have the access to glucose even if there is a low concentration in blood. Also liver, kidney, intestine, which is in a little different situation. It also uses most of fructose that is in diet. Around 80% is metabolized in intestine, just to mention. And pancreas, that is very important organ in respect to the glucose metabolism. It regulates, as we'll come later to that point. It also does not depend on insulin. Brain must have the access to glucose even if it's a very low concentration. However, skeletal muscle, adipose, heart, they strongly depend on the insulin. And intestine, that was already mentioned, like in case of fructose also, not only glucose, but fructose also does not depend on the intestine, on insulin. What is the dependence? Insulin increase the number of glucose transporters that are present in the plasma membranes. So that's the role of insulin. Insulin, we'll see similar role of insulin in other processes, but this is the way how it decreases the amount. This symbolically shown to just increase the number of glucose transporters, and that way allow to take more and more glucose. So that way we get glucose into the cell. And now with any cell of our organism, it is immediately converted. It is immediately, we call it trapped, because glucose can pass through the transporters, can transport glucose, but free glucose. If it is phosphorylated, which is an ester at carbon number 6, and this process is hydrolyzed by hexokinases, which are present in all cells, and glucokinase with a characteristic for liver. And I will tell few differences between them, and it shows also the logic of that metabolic differentiation. So nonetheless, the process is just the phosphorylation of glucose at carbon number 6, producing glucose 6-phosphate, G6P, which is the major intracellular form of glucose labeled for any metabolic process running in the cell. It is a process with the decrease in energy due to the hydrolysis of ATP, which is very important because this process is easily going from free to phosphorylated form. So hexo and glucokinases differ with their Km so the affinity for the glucose. Basically, hexokinases has a much lower Km. They are very efficient in the low concentration. They have roughly around below even 1 millimolar Km. So they very quickly take glucose even at low concentration, which is very important because, as you could have seen, in many cases it does not depend on insulin, and many tissues must have the access to glucose, like brain, red blood cells, etc. They are all inhibited by the product. So we don't take too much glucose. We take glucose in amount that we need. And then we produce glucose 6-phosphate. And if we don't use glucose 6-phosphate as a central metabolic form of glucose in the cell, we inhibit. Enzyme is inhibited and stop converting glucose. So glucose then can escape because it depends only on the gradient, where it concentration is higher. Now, hexokinase, this is called hexokinase D, or more commonly glucokinase, has much higher Km, a few millimolar. Optimal activity is around 10 millimolar. So it is not inhibited by glucose. So the role is completely different, and it shows also the role of liver in that process. The role of hexokinase D is to regulate the blood glucose level. If there is too much glucose, then it is taken by the liver. And this is very important role. Therefore, it does not compete if there is a low concentration of glucose, because it is inefficient. And then liver does not take the role of liver if there is a very low concentration of glucose in blood, is just to produce glucose that we are going to talk a little later. So this is very important process in which hexokinase, they participate in general overall strategy of carbohydrate metabolism. Now, what happens once this G6P is formed in the cell? What can happen with it? There are few major processes we are going in a moment discuss in more details. First of all, there is glycolysis. I put it centrally. It is in every tissue active. This is responsible for generation of energy. Very easy, very quick, very convenient way. Every organism loves glucose for that reason, because it can easily get energy. Then there is a mention already, phosphopentose pathway. It also runs in all tissue, but the aim, the goal of that oxidation, as it was mentioned a moment ago, oxidation, alternative oxidation of glucose to glycolysis, is completely different, and we are going to talk in details when we focus on phosphopentose pathway. Then, as mentioned already, organism, and I will try to convince you about that, organism must be able to produce glucose. Not only to use glucose, but produce glucose. And why that will be discussed later on, because as I mentioned, we cannot decrease the level of glucose in blood below the certain concentration. Otherwise, we are dead. So this is important issue. I will explain why, but I will remind you why. However, for that purpose, organism body is equipped with the possibility, the process of synthesis from non-glucogenic structures. So de novo, as we can say, and the liver is the major site. Intestine participate very little and for local need. Kidney, cortex participate in the, medulla participate in the only limited synthesis. The major from the overall body interest, there is the liver that produces glucose. So it's gluconeogenesis. We are going to talk separately about that. Finally, glucose can be converted to many various derivatives, glucuronides, sugar interconversions, etc., pentoses to synthesize nucleic acid, etc. So that's just a mention, not to forget. We are not going to focus in detail because these are quite complex, often processes. But there is only one thing which I have to mention also here in the beginning. As I mentioned formerly, glucose, the excess of glucose can be stored in form of glycogen. We are going to discuss it in more details, not this week, but probably next week, but next Wednesday. However, the process of release of glucose from glycogen is called glycogenolysis or glycogen degradation, while the process of synthesis is glycogen genesis or synthesis of glycogen. And there are only two organs that have, two tissues that have the substantial amount of glycogen. It's liver and muscle. Muscle is more selfish. It's only for muscle use. Glycogen, liver is serving the whole body that way from the storage form of glucose, which is glycogen. So this is the overview how those processes are. If I may just please for a second. Maćku, mam wykład w tej chwili. Zadzwonimy, tak, ja dzisiaj zadzwonię. Dobra, pa. Sorry, but Dean was calling to me. I don't know what's going on. Okay, so that's the overview of the processes of carbohydrate metabolism. Now let's go to glycolysis. Glycolysis, as you've already mentioned, it's the chemistry of glucose breakdown. There are some processes, 10 processes. They are fairly simple, but very important, crucial for the form of life as we know. 10 reactions, as I already mentioned. We will meet them. We'll talk a bit. I will not focus on some details. And this is maybe a good point to mention that I will try to avoid talking in too details about processes or the problems that are simple, but I will try to focus on those that are, from my point of view, require some more explanation, discussion, and thinking. The very important is that they are, of course, enzymatic, so they rely on enzymatic activity of enzymes that are able to convert intermediates. Finally, this is very crucial. This does not depend on oxygen. As you will see, this is very important. And this is a larger flux of carbon in cells. So if we consider the use of carbon, the life as we know is based on carbon, the largest flux of carbon in cells is in glycolysis. Now, to continue, it's a fundamental source of metabolic energy and occurs in all cells. Then it is a primary source for some tissues. In brain, for example, it may be supplementary, but the major pathway through which brain begins to get energy is glycolysis. Erythrocytes fully rely on glycolysis. They cannot get energy from other source. You already know very well the hemoglobin story, and erythrocyte is full of oxygen but doesn't have mitochondria. Therefore, it cannot, for that reason that it's full of oxygen, therefore it has to have the access to energy, and the glycolysis is the solely source of energy. Also, sperm cells and renal medulla, for example, also lenses and some other also depend on that. Finally, it's a source of carbon. That's very important because so far we're talking about the gaining of energy, but it also is a source of some intermediates which are mentioned already formerly as a backbone for lipids, fatty acid, nucleotides, cofactors, amino, etc. So that's a very important role also. Anabolic, because so far we are talking about a catabolic role, delivering energy, but here we can see that it's also some structures, intermediate structures are used for anabolic process. Okay, if we look into glycolysis overview, we can split it into two phases. First phase is the investment. Like in every business, first you have to invest, and then you can gain if this investment is reasonable. With no doubt, glycolysis was the very great investment in the history of life. So this in the first phase, we invest putting some energy into the molecule, and then we get more energy. So in general, finally, we get net 2 ATP molecules from 1 glucose molecules oxidized or metabolized through glycolysis, those 10 reactions. It's perhaps you can say, okay, it's not so much. Yeah, I know, it's not so much, but it is. It's the only, maybe when you discuss the oxidative phosphorylation and the overview of metabolism, you were able to notice that this is the only structure that can deliver energy in organism without using oxygen. So that's why it is so important. This is one of the reasons why we cannot fully get rid of glucose. Okay, I will not focus on that. Briefly, this is an overview how we go from glucose. You already discussed the hexokinase or glucokinase, doesn't matter which one, glucose 6-phosphate. Then it's an easy high isomerization by phosphoglucomutase. We converted aldose to ketose, it's fructose 6-phosphate, the isomer of that glucose 6-phosphate, and then comes the enzyme that we call phosphofructokinase 1. Rather remember that name. I will come back later, not so late, but I will come back later to it. Phosphofructokinase, and I will tell you why. And this will come also later when we focus on that. This is a key enzyme of glycolysis. So once fructose 6-phosphate is phosphorylated, this is one investment. This is second investment, the energy that we have to invest to convert this structure to fructose biphosphate. 1,6-fructose biphosphate. In this structure, once it is reached, once we produce it, there is no practically significant control. It can run and flux through the pathway. And the first step is just to split into two trioses, which one is glyceraldehyde 3-phosphate and second is dihydroxyacetone phosphate. You remember that the glyceraldehyde 3-phosphate and dihydroxyacetone phosphate are the simplest monosaccharides, trioses, the simplest because they have formyl group and hydroxyl group, or keto group and hydroxyl group. So these are the simplest monosaccharide. So they are split, and now this is the same shown by structure. So I recommend you to look through that if you want to understand a little bit more from the chemical point of view. So this is how it goes at the level of structures, and then those trioses, one of those trioses, as you could have seen here, we have true trioses, but they can be easily converted one into other. So as we are using that one for further processes, so the decreasing concentration in the system of glyceraldehyde 3-phosphate is due to this causing to the conversion of dihydroxyacetone phosphate into glyceraldehyde triphosphate. So finally, the molecule that is further conversion is glyceraldehyde triphosphate. And glyceraldehyde triphosphate now undergoes oxidation. This is the very important moment, because this oxidation, without using oxygen, produces the high energy compound, the compound that easily want to get rid of phosphate, we call it, want to get rid of that phosphate. It forms so-called high energy bond. So let's look at that in a little more details, because that's the beginning of the gaining of energy. So far we invested 2 ATP per glucose unit. Now we want to get back that, or even reach some more. So that's the beginning, and let's go on, look at this process. This is the only process I would like you to look in more details regarding glycolysis. This is the oxidation of glyceraldehyde triphosphate into 1,3-biphosphoglycerate. And these are two ingenious moments. The enzyme is called glyceraldehyde triphosphate dehydrogenase, responsible for that. And now this enzyme, this enzyme binds, it has a thiol group in its active site, binds this aldehyde. The aldehydes have very big affinity into the thiol groups, and they form here derivative that is of hydroxyl structure. So it is a kind of reduction, because it is a ACE H group. Now this hydrogen is transferred onto oxygen, this bond breaks and forms link with the enzyme. Now, this is the oxidation. You know very well, and I put there the structures, or they are involved in the process, but I am not going to focus on them. This NAD, because it was already discussed formally. So NAD takes 2 hydrogens, and it forms immediately carboxyl group derivative, because it is typical hydroxyl group derivative. But it is not, and if it is bind to hydroxyl group, it would be ester, but it's thioester because it's sulfhydryl group, which is alternative to hydroxyl group, but a little different due to the difference between sulfur and oxygen. So this is typical thioester. And the ingenious second thing is that there is no hydrolysis, because if we would hydrolyze that, we would not get high energy compound. And this is phosphorylasis. This is the ingenuity in that process, that we use phosphate, inorganic phosphate, to split that bond, and that way we are getting the anhydride. And perhaps you remember when you talk about the high energy compound, and anhydrides are the high energy compound, the energy of high potential of release of phosphate. It wants to get rid of phosphate because they are negative charges and located in a close contact with oxygens. This was already discussed. This is one of the highest energy compound in our organism. And in effect of that, due to this use of phosphate, simple phosphate and phosphorylasis, we get the high energy compound. And that is the reason why this is this reaction, 1,3-biphosphoglycerate. And now this 1,3-biphosphoglycerate can transfer phosphate onto ADP, like it was in oxidative phosphorylation, that phosphate binds to ADP, but here it is transferred from high energy compound onto the ADP and forms ATP. And finally, this produces 3-phosphoglycerate, which is a low energy compound, but it undergoes the further conversion. Before I go further, I want to come back for a moment. Here you see that we have two because glucose is 6 carbon, and this is triose, so 3 carbon, and they were split into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. But as glyceraldehyde 3-phosphate is used further, then, as I mentioned, dihydroxyacetone phosphate is converted to glyceraldehyde 3-phosphate, and now we have to multiply by 2 because there are 2 trioses out of glucose that are converted that way. So in that process, that it's substrate level phosphorylation number 1, we get from 2 ADP to ATPs. So we already are net zero because we invested 2 ATP and now we gain 2 ATP, but that's not the end. We come back to that process, which is triphosphoglycerate, which is at first almost useless, but due to the certain important processes, and especially the enolase and substrate level phosphorylation number 2 pyruvate kinase, it is again converted to very high energy compound. And let's come back here. We were here, 3-phosphoglycerate. 3-phosphoglycerate is converted by isomerase, triose isomerase into 2-phosphoglycerate, and 2-phosphoglycerate is now by enolase converted to phosphoenol pyruvate. I have to come back because this was here shown. 2-phosphoglycerate is converted to phosphoenol pyruvate, and again because we have 2 trioses, so we have 2 such structures, and enolase converts to the enophosphate, which is very high energy compound, and this high energy compound, even of higher energy than the 1,3-biphosphoglycerate, transfers phosphate, want to get rid of that. Phosphate is very unstable, so it transfers onto ADP, produces ATP, but there are 2 such molecules, so we have 2 ATP. And finally, if we look into the whole process, first of all, we produce 2 NADH because we oxidize glyceraldehyde 3-phosphate 2 molecules, and then net we use 2 ATP, and then we produce twice 2 ATPs, so the net is 2 ATP. And sorry, these 2 should not be here, of course. Okay, it's okay, sorry. 2 pyruvate, these are 2 trioses. Pyruvate is keto acid, as you already heard about it quite a lot. Pyruvate is one of the most important keto acids in our organism. So the overall effect of the process is that we got from glucose 2 pyruvates and 2 ATPs, oxidizing glucose without oxygen. So that's something to remember. The decrease in free energy is around over 80 kilojoules per mole, so it's quite a convenient and substantial decrease, so the process goes on easily. Now, pyruvate, that is the product of glycolysis. So oxidation of glucose without oxygen, 2 pyruvate, 2 triose, 2 keto acids from 1 glucose. If you compare the structure of glucose and compare the structure of pyruvate, you will see that this is the difference in oxidation of the compound. And this is why we call it oxidation of glucose, and NADH is the product, one of products, as we could have seen here, NADH. And of course, ATP as a reason why it goes on. Now, pyruvate can be reduced to lactate in so-called anaerobic glycolysis. So if glycolysis runs in the conditions when we don't have access to oxygen, then we call it an anaerobic glycolysis, and in this case, pyruvate, the final product of the glycolysis, is converted to lactate, and lactate is the end product. I will focus on that in a moment. Can be oxidized further, can be carboxylated and transaminated. That all will be discussed, but let's begin with the reduction. If you look into the glycolysis and conversion of glucose to pyruvate, that's something that goes in yeast. In yeast, in anaerobic conditions, yeast produce ethanol that we like, sometimes in form of beer, wine, if somebody likes vodka, etc. So, but luckily we don't produce, in pathological states, sometimes we see that production, but very, very little. That's not the way how it goes in our organism. It is a yeast are the major organism that produce it very efficiently. Now, glucose is converted either from pyruvate. It's oxidized in aerobic conditions, etc. That will be discussed separately in a moment. But if there is anaerobic condition, it produces lactate. Lactate synthesis is taking place vigorously in a very heavy loaded with the work contracting muscles, skeletal muscle, in red blood cells, of course, in some other cells, as I said, lenses and in renal medulla, etc. So this is the process that may go on in our organism, but as you will see, this is not very optimistic to rely only on that process, because it produces not so much energy. 2 ATP molecules per glucose. You will see how many molecules of ATP we can reach oxidizing glucose to carbon dioxide and water, from which autotrophs produce glucose, by the way. But autotrophs use the energy of sun. Okay, now, if we compare the aerobic metabolism of glucose, so producing water and carbon dioxide, versus anaerobic glycolysis, generally under normal respiring conditions, when we have access to oxygen, sufficient enough oxygen, the pyruvate is oxidized further, and it requires mitochondria for further process. That was already discussed formerly in respect to oxidative phosphorylation. However, under hypoxic conditions like an overload, active muscle that produces so-called oxygen debt, they are not perfused so quickly with oxygen, but they have to do their work. So they have to use the glycolysis and produce much less ATP, but produce ATP. In the cells lacking mitochondrial lens, cornea, erythrocytes that were mentioned many times, pyruvate is reduced to lactate and secreted from those cells. This does not resolve totally the problem, what happens further with that, but that will come later on. But nonetheless, for a given cell, like a given red blood cell, it finally produces lactate. Why lactate? Why it is the solution for its need? If you look into the structure, this reduction to lactate upon anaerobic glycolysis, this pyruvate is converted to lactate, as you see, using NADH as a source of the hydrogens. And that way, it restores NAD that can be used for further glycolysis or further turn or round of glycolysis, and lactate in this case, here reduced ketone to hydroxyl is hydrox acid is the lactate that is then secreted from the given cell. From at the body level, it is still something to happen with it, but in the cell level that uses that process, it restores an AD that is necessary to run the glycolysis, and that is shown here. The glucose is converted to pyruvate and uses to an ADH, and then due to the pyruvate reduced to lactate, it is balanced according to number of carbons, sorry, it restores an AD plus. So oxidized form of this coenzyme, adenine nicotinamide nucleotide, and due to that it can run glycolysis further. So an effect, as you remember, is ATP in each case. So that way, if we get rid of lactate from a cell, the cell gets energy. But for the body, it's not the end of the story, and that will come later on in respect to lactate. But for a given cell, it is a way. So anaerobic glycolysis, where we don't have access to oxygen, and we cannot use quickly oxygen, there is 2 ATP molecules and 2 lactate, which is further converted. And you could have expected that, as you could have seen already, the role of liver, that it will happen in liver, but that will come later. In case of aerobic conditions, and an ADH is not reoxidized, we cannot go that way. We cannot, because we want to use pyruvate and oxidize it further in mitochondria. So this process does not restore NAD+. And in these circumstances, if we use further oxidation of pyruvate, then we have to do something with this NADH. And the only way how to do is to transfer hydrogen to mitochondria, and that will come later. I will discuss it later. So this NADH must be reoxidized in mitochondria, but how it happens, we'll talk a little later. So now let's get back briefly to glycolysis, just to discuss how it is regulated, because it's a very important aspect of overall metabolism, not only of carbohydrate, but also of the energy in our organ. So here you see the glucose, glucose 6, all those intermediates to the lactate as the end. If we look into profile, energy profile, you will see that there are few processes that run with the high decrease in energy. And this is the first reaction, the phosphorylation, when we put the energy or we invest the energy. Second is fructose 6-phosphate phosphorylation. So the hexokinase, phosphofructokinase, you will get familiar with that name, I hope. And finally, the last, the pyruvate kinase, so conversion of phosphoenol pyruvate to pyruvate. Those three enzymes run the processes that go with a very high decrease in energy. So they are, this is not easy to revert them. And these are very good enzymes to control the flux through the whole process. That's the major strategy of the regulatory processes. So now I put those enzymes, hexokinase, phosphofructokinase, and pyruvate kinase in appropriate place. Now first, as you already heard, which I mentioned at the very beginning, hexokinase is inhibited by glucose 6-phosphate. If there is the high concentration of glucose 6-phosphate, it stops the further phosphorylation of glucose. However, I have to put here the red line, because if you will return to the slide that I showed you at the beginning, when glucose enters cells and is converted to glucose 6-phosphate, I showed you that this glucose 6-phosphate is the structure that is used for various purposes. Glycolysis is not the only way of usage of glucose 6-phosphate. Therefore, we sometimes say that this is a glycolytic enzyme, But on the other hand, we have to be aware that this role of hexokinase is broader. The role of glucose and hexokinase is broader because it traps glucose as a phosphorylated structure in the cell and does not let it out, but does not necessarily is used in glycolysis. However, if this will be used, the easy converted form isomer of the fructose, glucose 6-phosphate, fructose 6-phosphate, is going to be oxidized by phosphorylated by phosphofructokinase 1, this will go quickly up to the end. All those reactions go only because accumulation of the substrate, which is converted by the further enzyme into the product, etc. And product is the substrate for the next stage, etc., etc. This is the key enzyme of glycolysis. Phosphofructokinase 1, and phosphofructokinase 1 is converted is regulated by AMP. So if there is a high concentration of AMP, which means that is a low concentration of ATP, so we have the decrease in energy. And if it is so, then of course this stimulates the activity of phosphofructokinase because we want to increase the concentration of ATP, so we have to run glycolysis to produce ATP, which is understandable. And the second is fructose 2,6 biphosphate. That's right now you don't understand it completely, but I will focus on that in a moment because it's a very, very important molecule. Then there are important inhibitors. Citrate, which is the overall, you will understand that more when we discuss more about lipids as well. This is the meaning, the level of citrate means to signals the level of energy in the cell. If there is a high citrate concentration, it means that we have to slow down certain processes, catabolic processes. So that's why it also stops the key enzyme of glycolysis. ATP is reverse of that. That's what I told about AMP, quite clear. And the proton is also understandable because usually we have the acidification or high concentration of proton that already discussed in oxidative phosphorylation whenever we have the very quick metabolism. Quick metabolism acidifies. Why? You could have seen lactate. Lactic acid is the derivative of acetic acid. Acetic acid is quite a sour structure, and this is acid. So every metabolism produces proton. Therefore, those three as a meaning of the satisfactory level of the metabolism, high energy signals inhibition of the phosphofructokinase. And the only other enzyme is pyruvate kinase, is the end story. That is, as you see, it is activated by fructose biphosphate. Fructose biphosphate is product of phosphofructokinase 1. This is done the way that if we want to go quickly through and have a flux through the whole pathway, then we have to prepare every enzyme. Those enzymes have just run if they have enough substrate, then go on. But this is the end process, and we don't know what is going on to happen with pyruvate. Therefore, it has to quickly let the pyruvate be processed further. So this is the pyruvate kinase is activated by the product of phosphofructokinase 1, which adjusts the flux through the process, and also it's inhibited by the ATP, which is quite understandable because if we have enough energy, we don't want to use that without any reason, the glucose and the glycolytic pathway, and then alanine, which is the product of the transamination of pyruvate. That is the relation between the protein and between the metabolism of carbohydrates. So it may be right now not understandable, but this is also the signal from metabolism that says, okay, stop, don't go so quick because there is not need to now oxidize glucose. If we still see the use of fructose 6-phosphate, this is phosphofructokinase, the enzyme. When there is low ATP and high AMP, this is just to add to that point. If it is low ATP, it goes quickly, high AMP. If it's high AMP ATP, this is the enzyme that is very sensitive, allosteric enzyme. This is allosteric enzyme, very sensitive to that. Just to show. And another molecule, we are talking about key enzyme of glycolysis. Another is 1,3 fructose 2,6 biphosphate. 2,6, not 1. 1 would be here. 1,6 would be here. 2,6 is here. This is carbon number 2. This is carbon number 1. Because this fructose is the ketose. And due to that, in Haworth projection, this is carbon number 2. And this is furan structure, not the pyran structure. So this is very important structure. I will come to that later. You right now see that molecule and you have to just understand that this is one of the most important regulator of carbohydrate and not only carbohydrate energy metabolism in our organism. You see how strong it is. If we have 0, no fructose 2,6 biphosphate, phosphofructokinase run like the typical Michaelis-Menten enzyme. Once we have the very slow concentration, you see what's going on, it speeds up significantly. This is an allosteric enzyme, like hemoglobin, these are typical Michaelis-Menten. This is 0.11, and here you see that it can also overcome the ATP, because sometimes we may need those intermediates for other reasons as ATP. So if there is enough ATP, but we want glycolysis to run, F26P, that I showed you a moment ago, F26P is the molecule to overcome the ATP. And you see here in ATP depend on how concentrated, but even in higher concentration of ATP, even in 3 millimolar or 5 millimolar, still if we have the higher concentration can go on. So this is just to show you that phosphofructokinase 1 is key enzyme of glycolysis, and fructose 2,6 biphosphate is by all means the most important regulator of that process. That will come later on. This is once again to summarize, fructose 2,6 biphosphate is not a direct intermediate of glycolysis. Fructose 1,6 biphosphate is not fructose 2,6 biphosphate. Can be converted by phosphatase, respective phosphatase, back to fructose 6-phosphate, like fructose 1,6 biphosphate by respective phosphatase, but different phosphatase can be converted also to fructose 6-phosphate if you look into the pathway. It's an allosteric activator of liver phosphofructokinase 1, which intensifies glycolysis, but it's also very important. It inhibits the opposite process to synthesis of fructose 1,6 biphosphate, so the glycolytic intermediate, which will become clear and understandable when we discuss a gluconeogenesis, one of the processes in which glucose is synthesized in organism using the same reactions. So that will come later. So it's very, very important because it controls one of the crucial points in the metabolism of carbohydrates. Okay, so now this is the overview of that process. Okay, the very important is that ATP as an energy, of course, alanine, and because we are just here in the pyruvate kinase, we discussed phosphofructokinase, mentioned hexokinase, now pyruvate kinase, and here there is a little more a complex process because on one hand there is this reaction, and these are the inhibitors that are activated that was already shown on the former slide, but the activity of this enzyme, so the pyruvate kinase, depend on the phosphorylation. And you will understand that better when we discuss later on the problem of synthesis of glucose and regulation of the key points in glucose metabolism. And this depends on the glucose level. If there is a very high glucose level, then we dephosphorylate this enzyme, and this is active. But once it is low glucose level, then we want to speed up, we want to stop the process of the synthesis, the use of glucose. So we want to stop this enzyme, and then this undergoes phosphorylation and is less active. Sorry, this is a certain, here it should be less active here and more active here, so it just escaped me from that. Okay, so this is just the third enzyme, which you should be aware that is also activated more or less accordingly. I will come to that very briefly later. Now, oxidation of to acetyl-CoA. So the second fate of pyruvate. First is reduction. We try to explain, we discuss the regulation of glycolysis. Now it's oxidation to acetyl-CoA. And oxidation to acetyl-CoA is just pure pyruvate dehydrogenase. This is the major enzyme that is responsible for oxidation of pyruvate into the acetyl-CoA. This is the molecule that already discussed, I think, on the former lectures. This is used in the Krebs cycle, and here it's fully oxidized to carbon dioxide and water. That's why we say that glucose can be oxidized to carbon dioxide, as you see, and water, which is not shown on that slide, just to show the most important molecules in that panel. Now, this is the overview of the reaction. Now I have to focus for a moment about this reaction. So you see here, oxidative decarboxylation of pyruvate. The process is called oxidative phosphorylation, decarboxylation, because on one hand, the molecule will be decarboxylated, on the other hand, and we will release carbon dioxide, and on the other hand, this will be oxidized. So the keto group will be converted to acetyl group. And if it is converted to acetyl group, so it must be oxidized. So therefore, we call that process the oxidative decarboxylation. As we will also meet in our organism the reactions that are non-oxidative decarboxylation, but that's completely different story. This is the oxidation of alpha keto acid. Oxidative decarboxylation of alpha keto acid. This is alpha keto acid because alpha carbon is the carbon closest to the carboxyl group. So this is alpha carbon. Alpha, it's a keto, so it's alpha keto acid, and the general term for that is oxidative decarboxylation of alpha keto acid. We have more of them function in our metabolism, and one of those is pyruvate. Now, this is a very complex process, very important enzyme, which in fact is a complex enzyme. It's a complex of three enzymatic activities, three different kind of peptides, and I will show you in magnification. This is the overview, but I will show you how big it is in the moment. So this pyruvate is, first of all, requires three enzymatic activity and five coenzymes. Coenzyme A, NAD, which is already mentioned, discussed, diphosphotiamine. By the way, the diphosphotiamine was discovered, it's vitamin B1. NAD is derivative of vitamin B3. This is the derivative of vitamin B1. This is derivative FAD is the derivative vitamin B2. And this is vitamin B5, panthenol, phosphopantetotein. Pantene Provi, the company that produces shampoos, it's because they claim that they add the provitamin B5. Therefore, that's the name of that company. So we see that they are B1, B2, B3, B5, and lipoate, which is not a vitamin, strictly speaking, because it's synthesized in our organism. And now the theory of vitamins and the term vitamin was introduced in just to briefly mention, in 1911, so over 100 years ago. And this was the guy of the Polish origin, Casimir Funk, who was working in the world, in Swiss, in Britain, in the US, but he introduced studying that problem due to the certain pathology, but this is beyond the scope of that lecture. So, very complex, with the high energy decrease. This is very, very important to be aware of that. Now, look. This is the magnification of that enzyme, the complex. This is larger than ribosome. You heard about ribosomes, you know that machinery. The pyruvate dehydrogenase is higher, but larger than the ribosome. Now, briefly, we have to just go through. I won't focus on some, not necessarily this. I don't get into the details of mechanism, but the first enzyme is called, this is the decarboxylation. So the diaphospho or diphosphotiamine, the derivative of vitamin B1, participates in binding pyruvate to the first enzymatic activity, which is called pyruvate dehydrogenase. And in this enzyme, there is a decarboxylation, and upon decarboxylation, as you see here, is decarboxylation. It is bound more or less similar as in case of glyceraldehyde 3-phosphate dehydrogenase, which was discussed earlier. Now, this structure is with immediate oxidation transferred onto lipoic acid. This is lipoic acid, this is dihydrolipoic acid, so it's a reduction of that acid and one hydrogen, and there is carboxylate, the carboxyl group here formed. So this is an oxidation on the contact between first and second enzymatic activity, which is the dihydrolipoyl transacellulase. And then we have to restore, because we have, upon the transfer of acetyl group onto the coenzyme A, forming the thiol ester. This is the thiol ester. And these stars mean that I will show you something in a moment. This thiol ester is, of course, oxidized form of already this structure, and decarboxylated. So this is oxidative decarboxylation. We have the hydrogens on this coenzyme, but we have to restore the enzymatic activity in oxidized form, oxidized lipolysine, because it's bound to lysine in the polypeptide chain. And now it transfers onto FAD, and this is activity coenzyme of the third activity. This is dihydrolipoyl dehydrogenase, and then it is restored to active form that can take hydrogens from the next round or turn of the reaction. It transfers onto NAD, reduces NADH. So in effect, we are getting, I will just come back for a moment, it produces acetyl-CoA releases carbon dioxide, but produces an NADH. So these are two major products, NADH, acetyl-CoA, and carbon dioxide, which is not further used in that respect. So decarboxylation, oxidation, transesterification, those maybe it's minor. The very important is just to be aware that pyruvate is decarboxylated, oxidized, produces NADH, those hydrogens, and acetyl-CoA. I put their slides with the coenzyme A, but this was already discussed. I'm not going to focus on that. The advantage of that multi-enzyme, multi-activity complex is that intermediate does not dissociate from the complex. There's higher rate of this is very, very important enzyme. If there is the lack of TPP, of thiamine pyrophosphate diphosphothiamine, this B1 derivative thiamine, this leads to the so-called beriberi disease. But that's not today's story. Okay, so this is pyruvate, coenzyme A, and NAD, because the other coenzymes are restored during the process, produce acetyl, carbon dioxide is released, and NAD plus. The very, very important process of oxidation of pyruvate, the alternative way, not reduction to lactate, but oxidation. Now, I put two such slides. At this slide, if you look into your PDF form, that looks like that, but this for certain reason. On the second slide, you see the whole story of the picture, so otherwise you wouldn't see that. But why it is so? Because this is quite obvious that if we convert pyruvate to acetyl-CoA, and the product is NADH and acetyl-CoA, so both, if we have the high concentration of acetyl and or NADH and or NADH, they, by the feedback process, will stop the activity of the enzyme. On the other hand, if we have the high concentration of coenzyme A or NAD+, it will speed up the process of conversion of pyruvate, because this is the use of coenzyme A and conversion to the very important intermediate of Krebs cycle, so the substrate of Krebs cycle. So this is how it is regulated, but the process looks more complex. I won't focus on that because it is not the direct effect of that molecule. Those molecules affect the similarly, like pyruvate kinase, as you recall yourself, or look later on that slide, which was showing the phosphorylation of pyruvate kinase in respect to the need of the body. Always it is in respect to the need of the body. That will become clearer when we talk about this in more details in the next lecture. So acetyl-CoA or NADH will activate the kinase. Kinase phosphorylates, and this makes enzyme pyruvate dehydrogenase inactive. And on the other hand, when we need the energy, so this is in contractile muscle, calcium is speeding activity of phosphatase that will dephosphorylate or will stop the activity of kinase that phosphorylates, so it will increase the amount of the active fraction. And this will be the active enzyme that produces acetyl-CoA. So if we now compare the production of ATP and NADH, because I can refer to what you already discussed last time on the last lecture regarding the oxidative phosphorylation. So anaerobic glycolysis is something that we discussed a moment ago, and we summarized that the end effect is 2 ATP and 2 lactate. Let's forget about what happens with the lactate. We'll come back to that, but this does not change the calculation regarding the ATP. So 1 glucose of ATP via anaerobic glycolysis in red blood cells or lens or whatever gives, in muscles, gives only ATP. Anaerobic glycolysis produces pyruvate. So to that stage, it also gives 2 ATP analogously. But then this, you remember, NADH was not reactivated to NAD plus by reduction of pyruvate to lactate, because that process does not go on when we have the oxidative conditions. So in this case, we have to do something with NADH. And I mentioned you, it has to be transported to mitochondria. I will come to that in a moment. Now, 2-pyruvate, which are the product, will be further oxidized to acetyl-CoA. Now, every oxidation of every molecule of pyruvate to acetyl-CoA produces, as we have seen a moment ago, NADH. Now, if we have 2-pyruvate, we have 2 NADH molecules. Now, this happens, up to that moment, this happened in cytoplasm, but this happens in mitochondria. So you know now that every molecule of acetyl-CoA, if you don't remember right now, please recall yourself looking into the Krebs cycle, that every molecule of acetyl-CoA will be converted to carbon dioxide and water, giving 10 molecules of ATP. So if we have 2 acetyls from 2-pyruvate, we have 20. But those 5 more will come from the fact that pyruvate oxidizing to acetyl produces an NADH. And NADH, we have 2 NADH molecules. So these 2 NADH, what is going on? Christian, what is going on? Christian is asking something. Christian, Christian, what's going on?
I have no idea. If I ask something, I'm sorry.
Okay, sorry. It's not you, but someone was showing him that he needs the contact. Okay, is there anyone want to say something? Because if not, I am just close to the end, but let me finish that. Oh, Christian Głąbiński, are you here?
Yeah, my bad. I must have accidentally pressed request control.
No problem. So, okay. I'm just brief to the end. So, if we have 20 and we have 2 NADH, if you look how NADH is the first hydrogen reductant that transfers onto the first complex. So it goes through the whole, and for 1 NADH, we have 2.5 ATP molecules. Therefore, we have altogether 5 molecules of ATP. This comes from what you have already done. The pyruvate, including the oxidation to acetyl-CoA, give 25 ATPs. Now, this is another story which I will show in the very brief moment, but as it is an NADH, we can transfer hydrogens from cytoplasm to mitochondria very efficiently, and to produce 2 NADH molecules in mitochondria. So this will give 5, or we can convert less efficiently to produce FADH in mitochondria, and then it will give 1.5 for each FADH because it transfers onto succinyl dehydrogenase level on coenzyme Q, so it is a less proton transfer from matrix to the intermembrane space, and less efficiency in the ATP production. So either we can get 3 or 5. If we add together 25 plus 2 is 27, plus 3 is 30, or 5, 32. So we see here that we have minimum 15 to 16 times as much energy produced from the oxidation of glucose to carbon dioxide and water to carbon dioxide and water instead of oxidation without oxygen. But that's the only molecule, that glucose, that can deliver energy without oxygen. Here, it is a full oxidation, a full effect. So just briefly, I would focus on that. I would like to ask you to look why I said that NADH from cytoplasm is converted to FADH2 in mitochondria and transport onto electron transport chain on the complex number 2. But this is very important. Hydrogens from cytoplasm, from NADH, are transferred onto mitochondrial FAD and produces reduced FADH in mitochondria and restores NADH that can run glycolysis, like in case when we don't have oxygen, and lactate dehydrogenase reduces pyruvate to lactate. Or the most efficiently when NADH hydrogens from cytoplasm, the same as was mentioned here, are transferring onto the mitochondrial NADH. This is much more complex system. This requires one enzyme. This requires more enzymes. Sorry. This is more complex system, but nonetheless, we can transfer hydrogens from cytoplasm to mitochondria, and then it goes through the whole transfer electron transport chain in mitochondria. So it will give the full efficiency. So 2.5 per one molecule, 5.2. So you see that much more efficient, and that was why this oxygen became so important, because using oxygen we are getting much more energy from the same molecule, and some other molecules except of glucose cannot be oxidized without oxygen. So that's why oxygen is important, and we see here how it gives the effect. So I would like you to look at that briefly, and now of course I stop at that moment because we reached the end of the lecture, and I will talk about carboxylation, and then we go phosphate pathway, hexose phosphate pathway, and finally the gluconeogenesis. Next lecture. We meet at, I'm sorry, that's not my idea, but we have to, as I know, meet at 7:45, guys, yeah? Are you going to survive that? Hello?
It'll be very hard, but we'll survive.
Yeah, we are.
I know it's hard, but it's not my idea. I am also in the same situation as you. If you can meet later, we can meet later, but you have next classes, as I understand. So, hello, do you leave?
Yeah, we have a class right after.
Yeah. Okay. So you are not dead. Did you survive this lecture? If you have any comments, you can write to me. Okay? Okay, guys. See you on Friday.
Thank you.
Thank you.
Thank you very much.
Bye.
See you.
Bye.
Thank you.
Thank you.
Bye.
Thank you very much.
Bye.
Thank you. Bye bye.
Thank you.